The traditional textbook definition characterizes skeletal muscle as a specialized mechanical engine. Under this model, its utility is limited to locomotion, posture, and force generation. Over the last 2 decades, clinical research has identified a different reality. Skeletal muscle is the body's largest active endocrine organ. The early evidence for this came from the study of interleukin-6. Researchers observed that muscle fibers release IL-6 directly into the systemic circulation during exercise, without any preceding tissue injury or immune inflammation. Biologists termed this vast network of muscle-derived signals the myokinome. It is a specific repertoire of bioactive molecules synthesized and secreted by contracting fibers. While identified as soluble proteins, these exerkines include specialized lipids, metabolic intermediates, and encapsulated genetic material. This secretory network operates as a systemic integration hub. The molecules released by working muscle travel through the circulation to coordinate energy balance, tissue repair, and immune function across every major organ system. Through molecules like IL-6, physical movement initiates a biochemical conversation that extends far beyond the muscle fiber itself. Early models of exercise physiology assumed that muscle tissue produced perhaps a few dozen systemic regulatory factors. High-resolution mass spectrometry of human and murine cells proved those estimates were low. We now know the secretome contains thousands of unique proteins. Proteomic profiling maps 5,710 unique proteins in the murine secretome and 3,285 in the human secretome. An 80% intersection exists between the two, showing massive evolutionary conservation. Contraction specifically alters a distinct 10 to 15% subset of this network. Currently, biologists have fully characterized the biological functions of only about 5% of these contraction-induced myokines. The exercise secretome is a programmed communication array where 95% of the specific messages have not yet been characterized. This volume of secretion introduces a logistical paradox. Many of the newly identified exerkines lack the standard secretory codes required to exit a cell. Traditional secretion uses the classical pathway. Coded proteins route from the endoplasmic reticulum through the Golgi apparatus and out via exocytosis. Factors like myostatin use this route. Unconventional secretion bypasses the Golgi entirely, using direct lysosomal translocation to push uncoded proteins to the membrane. Muscle tissue upregulates these pathways to manage severe metabolic stress, ensuring massive volumes of exerkines deploy without bottlenecking the classical secretory apparatus. A third release mechanism is ectodomain shedding. Here, membrane-bound precursors like fractalkine are enzymatically cleaved, literally sheared off into the systemic circulation. Skeletal muscle maintains a variety of these exit routes to guarantee immediate signal transmission during physical exertion. Releasing the signal is only half the problem. Soluble proteins face immediate degradation from circulating blood proteases the second they enter the bloodstream. To protect sensitive cargo, muscle packages its secretome into extracellular vesicles. These lipid bilayer transports shield exerkines from enzymatic breakdown and extend their systemic half-life. These fall into 2 distinct categories. Exosomes are small structures assembled inside multivesicular bodies, while larger ectosomes are created by direct outward budding from the cell surface. The contents of these vesicles are not random. Muscle relies on specific RNA-binding proteins to actively select and pack highly specialized molecular cargo. The most critical payload inside these structures are myomeres, short, non-coding, muscle-specific microRNAs, like miR1 and miR206. A pie chart of their regulatory influence shows microRNAs drive 70% of the systemic effect by degrading target RNA, while proteins account for only 10%. These muscle-derived vesicles provide a specialized mechanism for a mechanical tissue to remotely modify the genetic behavior of distant organs. The primary targets for these payloads are highly specific. The muscle-brain axis regulates exercise-induced neurogenesis and cognitive resilience. Specialized myokines like irisin and cathepsin B possess the exact structural properties required to breach the highly restrictive blood-brain barrier. Once inside the brain, irisin upregulates the expression of brain-derived neurotrophic factor, or BDNF, inside the hippocampus. This localized chemical trigger is the primary biological driver for neuroplasticity and memory retention. In the metabolic system, the muscle adipose axis dictates systemic energy partitioning and whole-body insulin sensitivity. This axis regulates the browning of white adipose tissue, a targeted change converting energy-storing white fat cells into energy-burning beige fat cells. Muscle-derived vesicles deliver miR1 directly to these white adipocytes. As the fat cells absorb the vesicle, the microRNA alters local lipolytic pathways and induces thermogenic genes like UCP1. Skeletal muscle operates as a central command architecture, improving neurological hardware in the brain while modifying the metabolic software in adipose tissue. The precision of this network is most evident in the muscle-gut-pancreas axis, which functions as a biochemical thermostat for blood glucose regulation. During severe contraction, systemic IL-6 releases into the bloodstream, traveling to the intestines to target L-cell receptors. This forces the L cells to secrete GLP-1, a potent incretin hormone. GLP-1 immediately acts upon pancreatic beta cells, potentiating insulin secretion and promoting their long-term survival. By linking contraction to incretin release, skeletal muscle exercises has a direct upstream control over the pancreas to manage glycemic stability. If healthy muscle preserves systemic balance, compromised muscle actively dismantles it. A deteriorated myokinome produces a maladaptive secretome. This balance scale visualizes the chemical shift inherent to aging and sarcopenia. Protective factors like IGF-1 and IL-15 plummet, while muscle-destroying factors like myostatin surge, halting protein synthesis. In states of obesity and type 2 diabetes, insulin-resistant muscle cells begin to manufacture and release an altered profile of extracellular vesicles. Senescent muscle fibers pack these outgoing vesicles with senescence-associated microRNAs, such as miR34A. These vesicles circulate to adjacent healthy tissues and the pancreas. They discharge their cargo, actively spreading cellular dysfunction and exacerbating systemic insulin resistance. Conditions categorized as chronic inflammaging and metabolic syndrome are driven by the systemic corruption of the muscle's broadcast network. The exercise response is an unbroken, systemic dialogue between thousands of circulating molecules, transferring epigenetic instructions across organ boundaries. Clinical medicine is currently mapping these individual myocrine signatures. Soon, specific exercise protocols may be custom-tuned to elicit precise outputs, prescribing routines to maximize targeted neuroprotection or bone density. Treating skeletal muscle purely as a mechanical force generator ignores its primary biological function. Therefore, preserving our skeletal muscle mass ultimately maintains the complex endocrine and epigenetic integrity of the entire human organism.